Hello guys, today in this video we will discuss about the pathophysiology of celiac disease. Here you can see different parts of GIT like stomach, duodenum and jejunum. Here this is zoomed in view of jejunum. The outermost part is made up of enterocytes. Nutrients and food particles are absorbed by those cells. Just below the enterocytes, this layer is called lamina propria. The enterocytes are covered by mucus. As you know, small intestine forms crypts and villi. Here, these depressed areas are called crypts and these elevated areas are called villas. Enterocytes also have small finger-like projections, which are called microvilli. They increase the surface area of small intestine. Now, you must be knowing that the gastrointestinal tract is coated by a special type of antibody, that is IgA. It helps to defend against the mucosal infection. Lamina propria also contains different types of cells like antigen presenting cells. They work as a communicator between innate and acquired immune system. In some parts of small intestine, Pierce patches and mesenteric lymph nodes are present, which helps to activate the immune response. They contains naive T cells. Naive means which are not yet activated. Now come here. Gluten is digested by luminal and brush border enzymes into amino acid and peptides, including a 33 amino acid alpha gliadin peptide, which is resistant to gastric and pancreatic and small intestine proteases. Some of the gliadin peptides may induce the epithelial cells to express interleukin 15. As you know, interleukins are messenger molecules. Interleukins means between two leukins or lymphocytes. So, this interleukin 15 triggers activation and proliferation of CD8 positive intraepithelial lymphocytes. These lymphocytes express NKG2D, which is a natural killer cells marker and receptor for MICA molecules. Enterocytes that have been induced to express surface MICA molecules in response to stress are then attacked by NKG2D expressing intraepithelial lymphocytes. This will cause epithelial damage and may enhance the passage of other gliadin peptides into the lamina propria. Now this gliadin gets absorbed and converted to deaminated gliadin with the help of tissue transglutaminase. This deaminated gliadin picked up by antigen presenting cells. This gliadin peptide interact with HLA-DQ2 and HLA-DQ8 on antigen presenting cells. Now antigen presenting cells bring this deaminated gliadin to lymphoid organ and presents to naive T cells. This contact activates the naive T cells and naive T cells eventually converted to either CD8 positive T killer cells or CD4 positive T helper cells, depending on the cytokine produced. As we know that mature CD8 positive T killer or cytotoxic T cells release granzyme and perforin, which can destroy the enterocytes. On the other hand, CD4 positive T helper cells activate B cells. Those activated B cells are converted to plasma cells, and plasma cells produce antibodies. Now these antibodies are anti-gliadin antibodies. So as soon as they bind with gliadin peptide, there will be inflammation and tissue destruction. Here I would like to mention that plasma cells not only produce anti-gliadin antibodies, they also produce antibodies like anti-endomysium antibodies and anti-tissue transglutaminase. So every time we eat food containing gliadin, there will be inflammation and destruction of GIT lining mucosa. That's all guys. Please like, comment and share and don't forget to subscribe. Thank you.